Hello, in this video we will look at the plant hormone or phytohormone. So in this video we will just understand the overview of the plant hormones, and this is the series of the plant hormones, which that is the pathway of the plant hormone and the mode of action we will discuss in the later videos. So in this video we will discuss just the overview. So here is for example the plant which that is grow. Lateral growth due to the increase of the stem, and the vertical growth shows that the height of the plant, while here is the upper part is the green color shows that the leaf and stems, so that is the green leaf and stem. So the color of plant is due to the hormone, and the sunlight will be toward the sun is the tropic movement, positive or negative tropic movement is the away. So the apical part is also dominant due to the enzymatic activity, and root growth is also regulated by the hormones. So what is the hormone? Hormone in two types. Basically, hormone maintain and develop or growth of the plant, and this that is the regulators or inhibitors. It is two type. For example, here we will discuss about first is the auxin, indole acid. This auxin formula is look like this, and that is basically stimulate the apical dominance, and this also the cell enlargement. It means the cell enlargement means the wider of the cell, so that is why an apical dominance increase, while the root is we will also discuss here. But important thing is that here is the stem basically which wide the cell. It means the cell division in cambium as well as the enlargement of the cell. So the cambium are present between the xylem and phloem. So you can see in this diagram from left to right to show that the enlargement of the cell. And here the cutting of the wood or some causes the growth of the plant due to the production of auxin. And on the other hand, important thing is that here is you can see is the cambium ring will be active, and more cell divide in the cambium ring due to the accumulation of the indolestic acid or auxin. So here is when the low concentration of the auxin will promote the root growth, and the geotropism will be occur. It means that toward the gravity root will be moved. But when the higher concentration of the auxin will inhibit the growth of the root. So that is the important things. And one more thing we will discuss also, but here is the next we will discuss about the Gibraline. This gibralic acid is basically is the formula is look like this, and it can be produced from the fungal culture. So the fungal culture, and it is the part of every plant, even algae as well as the algae and basically fungi, and you can say that a bacteria is not contain Gibraline. So next here we will discuss about the effect on plant. So basically here is the auxin used for the cell division due to the accumulation of auxin and growth of the leaf, as well as the bud initiation, it means the bud production, as well as the cell division in meristem and cambium. Meristem mean that stem which that is able to divide the cell division. So the fruit growth as well as basically the parthenocarpy. Parthenocarpy is the growth of the fruit without seed formation, so without fertilization. On the other hand, here is the falling of the leaf is the abscission or the another basically is that the leaf senescence is inhibited by this hormone, and the break bud and seed dormancy. So it is bud dormancy and seed dormancy will be break, and bud initiation, shoot formation will be occur. And here is the auxin which that is promote the gibraline with the auxin to enlargement of the cell. So it means the collectively gibraline as well as the auxin is play important role to enlargement of the cell. So here is the red light during the daytime. It means the long day plant will be by the flowering, promote flowering. Why? Due to the red light promotion, which that increase the gibberellin. And when gibberellin will be decreased during the short day plant, for short day plant, so the flower will be promote. So this is the antagonistic part. And that on the other hand, next here we will discuss about the cytokinins. The cytokinins, the important is that their formula is look like this, and this is used for the cell division in apical meristem. It means the apical part of the plant, for example the apex of the plant, which that is the cell divide in apical meristem. Meristematic tissue is that which that is able to divide. So here is the initiation. It means the break bud dormancy and the growth of the leaf. On the other hand, that is the inhibition of the senescence and abscission. Abscission means the falling of the leaf due to the aging, and senescence is the basically is the falling of the leaf. So it will inhibit for the falling of the leaf and the growth of the fruit, as well as the cell division in cambium. And on the other hand, here is the auxin, as well as also the cytokinin, which that is promote the bud initiation, and indolestic acid will inhibit the abscission, as well as the fruit growth and the shoot growth, and the parthenocarp is also. And on the other hand, the one more thing is that the opening of the stomata also promote the lateral growth of the root, and the primary growth of the root will be inhibited by this cytokinin. So that means the primary growth will be inhibited and the secondary growth will be promoted. On the other hand, the seed dormancy is also break, so to produce the seed to embryo formation. So important thing is here is the abscisic acid we will discuss here is used for the abscission of the leaf. It means the leaf senescence. It means promote the aging of the leaf. Why? Because it is the excretophore is known as the leaf, which that is excrete the high metallic ion, as well as it is used for the closing of the stomata during the stress time, when the drought or the basically the water stress. So on the other hand, it is inhibit the root growth and promote the seed break dormancy. And on the other hand, the short day plant, which that is promote the flower, and the long day plant, which that is, for example, it is inhibit that flowering, antagonistic to gibberellin. Basically gibberellin is antagonistic. So next is the last one is the ethene. That is the ethylene, or we can say that the ethene. This is the very basically used for the ripening of the fruit and the break dormancy and stem growth. It means the stem will be wider and the growth of the root will be inhibited. So sorry for that, is the stem growth is basically inhibited and the root growth is also inhibited, and break dormancy of the bud and bud initiation is due to the ethene. And on the other hand, the pineapple plant, which that is promote the flowering of the pineapple due to the ethene. So ethene is used for the ripening of the fruit. When the ethene level will be high, so the fruit will become a ripened, so the ripening of the fruit. While during the decrease of the ethene, it means that during the developmental time the fruit will be developed and grow more. After the ethene will be produced more, it means the ripening will begin and complete ripening process will be occur. So that's all things was the overview plant hormones, that was the overview, and that's a phytohormone. So I hope you make sense about this. Next video we will discuss about the pathway.